Welcome to this public lecture on the theme of control, hosted by the History and Philosophy of Science program at the University of Melbourne. My name's Michael Arnold, and I am Professor and Head of Discipline of History and Philosophy of Science. The History and Philosophy of Science program began in 1946 and is one of the first departments of its kind in the world. Throughout that time, HPS has been located on Indigenous land. We acknowledge the elders and descendants of the Wurundjeri people who have been and are the custodians of these lands. Wherever you are in Australia on Zoom, you are on Indigenous land, the land that is the place of age-old ceremonies of celebration, initiation, and renewal. And that we acknowledge that the local Aboriginal peoples have had and continue to have a unique role in the life of these lands. We're privileged tonight to have Associate Professor Nicole Nelson, an internationally renowned researcher who will discuss the difficulty in understanding the results of replication experiments and the consequences that can have for researchers. I'm going to pass now to Professor Fiona Fiddler, a friend and colleague in History and Philosophy of Science and the head of the Replicats research program, who will introduce Nicole.
Hello, everyone. Thanks for joining us for this public lecture tonight. It's my great pleasure to introduce Associate Professor Nicole Nelson. Nicole is an Associate Professor in the Department of Medical History and Bioethics at the University of Wisconsin-Madison. She's also a member of the History of Science, Medicine and Technology program in the Department of History. And she's a member of the steering committee of the Holtz Centre for Science and Technology Studies. Nicole teaches courses in science and technology studies, biology and society, and history of science. She's won many awards for her undergraduate teaching, which won't surprise you when you hear her speak tonight. She's a collaborating editor for the Journal of Social Studies of Science, and she's also on the editorial board for the journal New Genetics and Society and the Journal of History of the Journal of the History of Biology. Her first book was called Model Behaviour, was published in 2018, and it looks at model organisms used in animal and human behaviour studies. In the few years since then, Nicole has turned her attention to the replicability or reproducibility crisis in some sciences, particularly in the biological sciences. And Nicole asked some very difficult questions, like what would the future look like if science was indeed fully open? How would we manage synthesis and how would we resolve debates? Tonight, however, she's talking about slightly more constrained questions about how to make sense of failed replication studies. And her talk tonight, in line with our SHAPS public lecture theme of control, is called Controlling the Interpretation of Replication Studies. Nicole?
Thank you so much, Fiona, for that wonderful introduction. I'm just going to flip on my slides now for you all. Okay, how is that looking for everybody? Okay. All right, so as Fiona says tonight, I'm going to be talking a little bit about this sort of a moment of ambiguity that happens when somebody attempts to do a replication study. And I'll start off by explaining this moment of ambiguity for you with this sort of theoretical example so that you can understand what it might be like to be in the laboratory, often as an early career researcher undertaking these types of experiments. Imagine that you're joining a lab and you've been asked to take over the project of a graduate student who has recently left. And that graduate student was trying to understand whether or not a new drug would slow the growth of cancer cells. So they've asked you to redo some of this graduate student's last steps and then carry on their research where they left off. So your first step is going to replicate the experiment that the original graduate student did, looking at 2 different treatments for the cells to see whether or not the cells in, say, the red dishes on the screen here grow more slowly than the cells in the control dishes, here represented in pink. And so you give a go to this experiment. You do exactly what they did, or so you think, based on what they wrote down in their notebooks, but you can't find a difference. The experiment fails to replicate. Now, in a kind of cartoon version of the scientific method, we might conclude that this means that we should reject the hypothesis that cancer cells grow more slowly when treated with this drug X in their growth medium. And if you Google the scientific method in Google Images search and look at what pops up, the image on your screen here is one of the first you'll see, this sort of multi-step process where you pose a question, make a hypothesis, you design an experiment, you test that hypothesis, and then you accept or reject the hypothesis. So we might say, you made a hypothesis that this drug X is going to slow the growth of cancer cells, and it didn't do that. So now is the time that we reject the hypothesis. But the scientific method is not quite so simple, because first we would have to determine if you actually did the experiment well. Did you do the experiment in the same way as your predecessor who found the original finding that drugs X does indeed slow cancer growth? And so if you look in the Google Images search for scientific method just a little bit further down the hierarchy, you'll find a sort of slightly different, a little bit more complexified tree of questions to ask here, where we construct a hypothesis, we test it with an experiment, but then we ask, is the procedure working? And then we might say that you should only reject the hypothesis that drug X does something to cancer cell growth if we've determined that the experiment is working. But then this leads us into another question. How do we know if the experiment is working? And when you think about that question, it quickly becomes really quite complex, because at the center of the experiment that I posed for you here, we're testing one question or one hypothesis, which is drug X is going to slow cancer cell growth. But along with that, we have to make all kinds of other assumptions in order to assume that the hypothesis will hold. We need to assume that the batch of drug X that we're using has not expired. We need to assume here that, the experimenter has correctly plated the cells, that they know how to put the cells in the growth medium so that the cells don't die. We need to assume that those cells haven't mutated, so that they haven't picked up or dropped some genes that would change their susceptibility to this particular drug. We need to know that the growth medium contains the same nutrients, maybe between the control and the drug, and between the replicate and the new study. So this is what's known in the history and philosophy of science as a Duhem-Quine thesis, that we can't test one hypothesis in isolation. We can only test bundles of hypotheses. We may be interested in this one question about whether or not drug X slows cancer cell growth, but we can't answer that question without making a whole bunch of other assumptions that we also need to test alongside it, because they're embedded in a complex experimental system. So this brings us to our problem for today, that when you conduct a replication experiment, the results, when you get a failure to replicate, are ambiguous, because one of many things could be going on. Any one of those different components of the experimental system that I pointed to could be malfunctioning. It could be something about the technique, the cells, the drug. Or the experimenter could be doing something wrong. Perhaps our novice experimenter who's joined the lab has been contaminating the plates. Or maybe the original member of the lab was wrong and their experiments just didn't work out for some reason. So all of these things are at play when a failure to replicate happens, and it's not clear which one of these assumptions we should prioritize over the other. And we do know from recent work testing the replicability of results in the biological sciences from the Cancer Reproducibility Project hosted by the US Center for Open Science, that many times the results that appear even in the published literature are wrong. They're not actually reproducible when we subject them to a replication experiment. So all of these potential answers are on the table when we encounter a failure to replicate. So the research question that I want to ask today is, how do people who fail to replicate a result interpret their experiment and then resolve the ambiguity of their situation? This is in fact a fairly old question in the field of science and technology studies. It can be dated back most in a rather famous work called Changing Order by Harry Collins, where he looks at scientists who are trying to replicate each other's results. What's different about what I'm going to present to you today is I'm going to focus uniquely on the experiences of early career researchers, particularly graduate students who are still learning to do the techniques while they're also performing replications. This gives us a particularly thorny situation because, as you will see in this talk, students often have reasons to doubt themselves, and they often default towards doubting themselves, which makes the interpretation of failure to replicate patterned in a particular way that prevents people from thinking that the original result might be wrong. So, to investigate the experiences of early career researchers, now about a year ago, I put together a study team composed of early career researchers, 2 undergraduates and 2 first-year medical students, who together with me did a survey and an interview-based study where we collected up survey responses from biomedical science graduate students at my home university, the University of Wisconsin-Madison. So we asked students in 4 different programs within the university if they would first fill out a survey for us. And that survey would look at whether or not they'd had an experience that looked something like a failure to replicate. And then if they had had one of those experiences, we then invited them to do an interview with us where we talked to them for half an hour about their experience. We asked them some open-ended questions like, what did you think was going to happen? Where did you get those assumptions? When it first went wrong, what did you think was the problem? What did you do next and who did you go to? How did the failure to replicate impact you, your career, your mental health? And through these open-ended qualitative interviews, we collected a set of rich stories from students about how these failures to replicate impact not only science, but the individuals doing the science, which in many cases is early career students. So here's what our study design looked like in the numbers. We first collected about 80, or exactly 80 survey responses from a total population of 126, and then conducted 37 paired interviews with those same students who participated in the study. One thing that you'll notice that's a little bit different about the people who participated in the survey versus the interviews is that we had many fewer women participating in the interviews. And I'll just note that we think that this is because we asked these people to participate in interviews in the early days of the pandemic, where a lot of women were experiencing an extra burden of care work at home, and so might not have had time to participate in that wing of our study. So keep in mind that the qualitative interviews largely reflect the experiences of men that we're going to be talking about today. So one of the first sets of questions that we asked them, and I recognize that this text is very small, don't realize, I'll read some of this out for you. One of the first sets of questions that we asked them is a series of hypothetical scenarios where we asked them if they'd experienced anything like what we described in the survey. The reason that we did this rather than asking them if they experienced a failure to replicate is precisely because of that ambiguity around what counts as a failure to replicate. How would they know if they'd failed to replicate the result or if they just messed up? So instead of saying, did you fail to replicate a result? We asked them a series of questions. Here's one of them. We asked them, have you ever experienced a situation where you were doing an experiment that gave you results were not consistent with what you expected? That's the very bottom bar down at this chart. And you can see that 98% of students said that yes, they had had at least one, and most of the time, several experiences like this. That's not too surprising. In science, if you're not getting results that are not consistent with what you've expected, you've probably chosen some very safe research questions. So we expected that most people were going to answer yes to this question. But then we posed a few different scenarios that we thought were a little bit more plausibly like failures to replicate. We asked them, have you experienced a situation where you yourself had done the experiment before, but got results that differed from your previous attempts? And what you'll see is that surprisingly, 84% of students said that they had had at least one, and sometimes several, experiences just like that. When we asked them whether or not they had failed to replicate something from the literature, 58% of students said that they'd had at least one experience where they tried to do a result that they'd seen in a publication and failed to replicate that result. So what we found here is that failures to replicate are an almost universal experience. Out of all of the students that we interviewed, that we surveyed, 79 out of 80 said that they had failed in some way to replicate their own finding, the finding of a colleague or somebody else in their laboratory, or something from the literature. So failures to replicate are not rare. In the biomedical sciences, at least, they're happening to students all the time. When we interviewed the students to find out more about what those experiences were like, we asked them to focus on one experience that they had had at some point in their scientific career. As you could see from the previous graph, many students reported having multiple experiences where they failed to replicate a result. And so what we did in our interviews is we asked them to take the experience that was most vivid in their minds, said to you, which one do you remember most clearly that had maybe the strongest impact on you? And then we stepped through that one experience in this semi-structured interview. Interestingly, you can see that when we posed the question to students that way, almost a third of them chose to describe an experience that happened to them before grad school, sometimes even in their undergraduate research careers, showing the impact of early experiences on students in terms of their identity formation as scientists. And that's something that I'll return to a little bit later in the talk. About a quarter of students chose to talk about failing to replicate their own work. About half of students chose to talk about failing to replicate their colleagues' work, a scenario that's quite similar to the theoretical one that I posed to you at the beginning of the talk. And then about another quarter talked about failing to replicate something from the literature. When we asked them how their problem resolved, we found that about half feel that they had solved whatever the issue was that came up in their replication. They'd identified what was different or what was malfunctioning in their experimental system. But the remaining half of students either shifted their research question or abandoned their project and did something entirely different because they were never fully able to resolve the ambiguity around what was going on. And then we asked them whether they had published the data that came out of these replication attempts. We found that a little more than a third had, but the majority of data from these experiences never made it into the published literature. When we asked students about their subjective interpretations of what was going on, that is, what they were thinking and feeling when they first encountered the failure to replicate, what we found was really striking because a situation that could have many possible outcomes was for students actually reduced to one fairly clear outcome, at least initially. 86% of students that we interviewed believed that they did something wrong. That was their first interpretation when they encountered a failure to replicate. And so all of the many-fold possibilities that I gave you in the beginning of the talk, a reagent could be wrong, the technique could be different, something varies between the experiments. Students initially dismissed all of those things and instead decided that it was their own lack of skill that they thought was the problem. That was the reason why they'd failed to replicate the result. And in the little quote bubbles here that on the side of the screen, I'm going to give you here some of the students' own words about what they thought was wrong in their situation that they described to us. One student put it quite succinctly and said, "I was fairly new to research. I just thought it was something that I was doing wrong." The student doesn't really specify what they thought they did wrong, but they're sure that in the vast landscape of things that could go wrong, the problem is located within them. A second student said, "My first thought was that I had done something wrong. So I was like, oh, I probably ordered the wrong sequence or I cloned the protein incorrectly," things like that. This student also locates the source of the problem in him or herself, but offers up a few potential examples of the types of things that they might have done wrong. Just for illustrative purposes, though, they weren't sure that they had ordered the wrong sequence, but they'd assumed that they made an error of this kind. And finally, the third student says, "It was just a different field for me too, which probably made me think that, oh, I'm just not knowledgeable enough to be able to do this because I didn't know much about neuroscience because my background was immunology." So this was a student that had done work in immunology as an undergraduate and then was moving into a lab that did neuroscience research and assumed that it was her lack of skill in this particular field that was the reason why the result had gone wrong. What's noteworthy here, though, is that this is a situation that's not unique to people switching fields. It's a situation faced by all early career researchers who are often unsure whether or not they haven't gained the skills yet to be able to properly execute techniques in their field, or if it's something wrong with the system itself. Now, these quotes portray these experiences as kind of mild, normal rites of passage that all grad students go through, of first assuming that they simply have not gained enough skill yet to make the experimental system work. But when we asked students about how these failures to replicate impacted them, what we found was the emotional impact of failing to replicate a result was in some cases quite strong. If you look at the histogram here, what we've reported is the way that students described their primary emotional response. So we took the students' own words and attempted to classify them into different kinds of ways that students might have reacted. What you'll see is that most common response here from students is self-doubt or a loss of confidence, which makes sense if they're assuming that they're at fault, that they experience a sort of hit to their scientific selves and wonder whether or not they're actually capable of doing research. Many people also reported feeling annoyed or frustrated, depressed or demotivated, and worried about wasting resources in their lab. The darker green colored bars in this response here represents people whose emotional responses were so strong that it interfered in some way with their day-to-day lives, meaning that they were no longer able to sleep properly, eat properly, perform their experiments. And to show you some of the strength of that impact, I'm going to give you a couple quotes now from these interviews from people who described their emotional response to their experience of failure to replicating as something that really deeply stayed with them. One student says, "I had anxiety and depression. It was not great. There were a couple of points in the middle where it really became functionally obstructive. Work productivity would just go down because of that. And then you just start assuming that the projects aren't going to work. You start assuming that the experiments don't work and aren't going to work. And then it becomes a vicious cycle." So what this student is describing here is the kind of process of feeling as though he's never going to be able to produce the result that he should be producing, and finding himself falling into depression because of that. Another student says, "I had constant headaches. My stress levels were very high. It made it hard to focus on school because I was constantly in the back of my mind trying to figure out what was wrong with my experiment, how to make it work. My sleeping became impaired because I was stressed, thinking about it all night and stuff." So again, what you can see here is that the student is working from the assumption that their experiment is wrong, not really entertaining the possibility that it might be the original result that's wrong. And so they're spending their time and emotional energy trying to troubleshoot what may not be a stable finding in the first place. A third quote from a student who had a similarly strong reaction to their failure to replicate. "I'm a person that really keeps a pretty level head, and I never really let my emotions get the best of me. I never thought that I would have said it, but looking back on it, I was actually depressed. I wasn't eating correctly. I wasn't sleeping correctly. Like I said, I'm a pretty strong person mentally, but I remember at one time after a meeting with my PI and being told what I already knew, but then also having my apparent inadequacy in my face, I just had to go to a single occupancy bathroom. I just got in the corner and broke down crying." This is a student who described the experience of his failure to replicate as so impactful on his experience as an early career researcher that he nearly dropped out of graduate school. And he wasn't the only one that told us in the course of these interviews that that experience of failing to replicate a result and doubting oneself was so impactful that they considered leaving the field. In fact, in my research team of 4 early career scholars, both of the medical students who were on my team told me that in their early days as undergraduates, they had both worked in laboratories, both failed to replicate results, both came to the conclusion that they were not cut out for science, and both decided to go to medical school instead. Now, that initial attribution that the results wouldn't appear because something that they were doing wrong is something that changed over time. Students began to reconsider that initial assumption of theirs as they continue to try and work and troubleshoot the problem. What students typically did when they encountered a failure to replicate is they started working around the edges, seeing if they could identify in what way they thought that they were wrong, working from that assumption that something that they were doing was bad. So students, for example, described repeating the experiment with new reagents, or repeating the experiment with somebody watching them, having somebody else in their laboratory, typically a more senior person, perform the experiment to see if they could get the results to work. Or sometimes they would introduce a new experiment alongside it, perhaps a control, to see if their antibody lacks specificity to be able to find what they did. What happened through the process of doing this troubleshooting is that many students' attributions of the problem changed. They started to doubt their self-doubt, and they started to doubt then the original results that they had found. In one student's words, the student recalls, "I did go through a lot of troubleshooting and reached out to other resources, but that still didn't work. I think with all of those elements combined, it made it harder and harder for me to believe in the validity of the original results that somebody else got." So the sometimes months-long process of trying to troubleshoot resulted in a bit of a shift of the way that students viewed the situation. What once seemed to be quite clearly their fault became something a little bit different. It became something where they could reopen the space of possibility and consider all those many things within the web of assumptions that were embedded in the experiment that might be wrong. Here's a diagram that shows you how it is that students' interpretations changed over time. In this diagram here on the left, what you're seeing is the initial attributions of the problem that the students made. And you can see that while a few students believed at first that their experimental results were due to bad reagents or failing equipment, or maybe some sort of difference between the original and new experiment, you can see that very large chunk of blue there represents the 86% of students who initially believed that it was their fault. What happens, though, is that through that troubleshooting process, the attributions diversify. So at the point at which we interviewed them, which for some students was mere months after the experience that they described, and for other students was several years, we asked them what they now believed to be the problem. What did they think at this point, with all of their new experiences, was the reason why they failed to find a new result? Here, what you'll see is quite interesting. We have more explanations, but the explanation has shifted so that now the largest category is that students believe that the original result or the hypothesis that they were working from was wrong. So students' attributions shifted over time from doubting themselves to doubting the premise that they were working with. Now, what happened is that this process of working through the experiment often brought students into conflict with their advisors. When students presented the results to their advisors, some found, those with a particular positive or supportive relationship, that their advisors confirmed their experimental skill and their scientific identity and said, "Yes, I believe that what you found is real, even if it conflicts with what we see in the literature." But what we saw more often was that advisors often believed that the experiment was just failing to work in the students' hands, meaning that students should be able to prove their experimental skill by getting an accepted result. And if they couldn't get the accepted result, it was evidence that they had not yet honed their craft finely enough to be able to produce trustworthy results at the bench. This trapped students in a moment where their own attributions shifted so that they now no longer believed the result that they were trying to work with, but they could not convince those around them, particularly their advisors, those who were gatekeepers to their career, that the results that they saw were no longer valid. Here's one student who describes this situation quite poignantly, who was in a position of deep frustration as now a senior graduate student about to graduate, who had been working with an experimental system for now several years and felt that he knew more about the experimental system than his advisor. And yet his advisor would not listen to him or believe in his expertise. The advisor continued to believe that the student had failed to acquire the skill to make this tricky technique work. The student says, "It always seems like the response to non-reproducing data is, well, let's see if we can figure out what you did wrong, as opposed to, well, let's see if we can really trust the previous data that we're comparing it to. Is there a reason why 70% is some magic number? Why? Why do we keep quoting 70%? Maybe the previous scientist had just as many problems as we did. Maybe he did 110 and 170, and he just picked 70 as the number to show. It's frustrating to have my experimental competency challenged every time it doesn't work, because I know more than the PI about how this is working." The system that the student was working with was a cancer model, a mouse model of cancer biology. And in the reported published literature, mice in this model were supposed to develop tumors 70% of the time. What the student found is that the response was actually quite variable. It did not work 70% of the time. Sometimes it was 10, sometimes it was 70. And he began to suspect that what had happened was actually a case of what's called selective reporting, where researchers might run several experiments and then pick the one that looks the best to publish. So when he says, maybe he did 110 and 170, what he means is maybe the original researcher also found the same amount of variation that he himself was finding in his experiments. Sometimes he'd get 10% of mice with tumors, sometimes 50, sometimes 70. And yet in the published paper, the researcher had chosen only to display that 70. The student's frustration, though, is that he was being held to the standard of the published literature, unable to escape the seeming authority of the paper that his advisor trusted over his own years-long experience working with this experimental system. All right, so to wrap up, I want to leave you with a couple major thoughts here about what we might be able to take away from this study that's focusing the spotlight on early career researchers. What we know from the literature already, and this study confirms it, is that replicating an existing finding is not something that's clear-cut because no two experiments are the same. There will never be two of the same experiments because they will, at minimum, always be conducted at a different time, sometimes by a different person, in a variable and changing world that never holds exactly still. This is an insight that Harry Collins came to in the 1980s and holds true today. It's the situation that gives us the ambiguity that the students we interviewed had to resolve. This lack of, or this ambiguity around the situation means that we could question the experiment, question the person, or question the original finding. All of those interpretations are on the table and possible, but what we observe through our data is that there's a pattern. The pattern is that students doubt themselves. They then move away from doubting themselves, but their advisors continue to doubt them as well. We didn't continue on to investigate what happens if they then tried to publish their results in the literature, but I think that we could hypothesize that one thing that might happen is that even if they manage to convince themselves and then their advisors, they face a further barrier in trying to convince the peer reviewers to believe in the contradictory result that they have produced. So this then, I think, gives us two things that we should worry about. The first is that we should be concerned that some proportion of true failures to replicate may be presently dismissed as student error. It is probably the case that beginning students are making mistakes. We should expect that because they're trainees, and we shouldn't overthrow well-developed scientific theories because a beginning graduate student found a contradictory result. Nobody is going to say that the theory of gravity doesn't replicate because an introductory physics student failed to find the right constant in their experiment. So it is true that some proportion of what's happening here likely is attributable to student error. But because of the ambiguity around these situations and the lack of measures for judging experimental competence, it's very difficult to tell how many of these things are truly student error or truly null findings. In fact, we find ourselves trapped in a problem where advisors and supervisors use the ability to get an expected result as a proxy for experimental skill. This has the effect of solidifying or reifying the results that are there in the literature because they're being used as the bar to judge whether somebody should be trusted as a credible experimenter. What this means is we might be slowing the pace at which the literature is corrected because we have no good way of determining whether a result is a true null finding or is a student error. And since the pattern is that students doubt themselves and are then doubted by their advisors, it seems likely to me that some proportion of true null findings are currently being dismissed as error and therefore not being reported or corrected in the literature. This is something that we should all be concerned about because it means that the incorrect results are no longer removed from the literature because they simply vanish into the sort of experience, the painful experience of learning to become a graduate student. The final thing that I think that we should care about is the impact of these failures to replicate on graduate students' mental health. Over the past 5 to 10 years, what we've seen in the literature on the mental health of trainees is that there is a crisis of mental health equal in proportion to the crisis of irreproducibility that we've seen in many scientific literatures. Some studies have found rates of mental illness amongst graduate students and PhD students especially that are as high as 4 to 6 times that of the general population. So if failures to replicate represent one point that's a trigger point in bringing on a mental health episode for graduate students, we should care about failures to replicate not only because they impact the scientific literature, but also because they impact the mental health of scientists. And they may, in fact, be a moment when some scientists who could be doing good science and potentially correcting the scientific record may choose to leave the field. So this is a problem that's not easily resolved because of this inherent ambiguity around the project of failures to replicate. But I think that we need to pay more attention to the trap, which is testing students by their ability to create an established result in the literature and recognize that the harms that this has both for the literature and for the mental health of the student. So thank you, and I will leave it there. Okay.
Okay.
There you go.
Thank you.
Oh,
Thank
sorry.
you. Stay here with me. Stay
Okay,
here with me.
okay.
You go back on this side.
All right.
Thanks very much for your excellent talk. There's a couple of questions in Q&A now, which we're going to answer live now, or Nicole's going to answer live now, but I'll just, while we're dealing with the first ones that have come in, please do type any other questions you have into Q&A. So the first question comes from Jason Chin, who asks, were there any conflicts with supervisors had to, in your experience in the interviews, were there any conflicts that these early career researchers had with supervisors had to do with not just with failing to replicate others, but failing to replicate the supervisor's previous findings? That would seem to create a certain type of friction.
Yeah, that's a really good question. And it's a perceptive one because it points to the importance of the power dynamics in the laboratory and the way in which laboratory supervisors, PIs, get invested in the things that they have already created. Now, something that's unique to the biomedical sciences is that typically PIs are no longer doing actual hands-on research by the time they reach the stage of directing their own independent laboratory. So they're usually not directing, or they're not replicating the direct work of the hands of their supervisor. But what they're very often replicating is the work of somebody who's worked in the laboratory of the supervisor, who the supervisor has sort of lent their credibility to by saying, yes, I believe the results, publishing the results with their name on it. And, undoing those results would undo not only a publication of the supervisors, but also sort of the supervisor's authority to judge who was a good knowledge producer. If the supervisor believed that his or her star postdoc was really good at doing a particular technique, and then a later student finds that the results from that postdoc are not actually trustworthy, then that also undoes their credibility as an advisor as well as a producer of science. So there's a couple layers there that would make people resistant to wanting to think about undoing the previous work, even if they hadn't done it with their own hands. And this is a situation that students find themselves in all the time. Most of the time, students told me that when they joined a new lab, they were given a test project, and that test project was to replicate the results from a standard technique that group used. So almost always that was a technique that somebody else had done before and the advisor had signed off on. So they were doing something that was kind of not the advisor's work, but his or her ownership, basically.
Thanks. Another question here from Matthew Page. He asks, what do you think led students to shift from blaming themselves to questioning the original finding? Do you have any sense of how long it took for that perspective to change? And is there anything that we can do to speed it up? I'm very interested in your answer to that last question. We have previously around here talked about issuing early career researchers with scripts that they can read out. But you've actually made videos of this, I know. So
well, I'm really pleased to say that I do have at least a hypothesis to test on that, because one of the things that we found from the qualitative interviews that we did is that there was a fairly marked difference between the students who assumed that the natural world was going to behave algorithmically and give them the same result every time, and the students who believed that the world was a kind of intrinsically variable or complex place. So if you'll remember in that kind of histogram of emotional responses that I showed you, there was actually a fairly large chunk of people who described their emotional response as indifference or lack of surprise. And if we drilled down into those students' experiences to see what was unique about that particular group of people that weren't very surprised, what we found is that those people had been primed in some way to expect that either the published literature wouldn't replicate, or that the natural world was complex and variable. So this leads us to hypothesize that one way that we might be able to intervene on this process and kind of shrink the time in which it takes students to open up the space of the possible explanations they're willing to consider is by maybe doing some sort of psychoeducational intervention where we talk about the stability of the literature and the reproducibility crisis. We talk about the many ways in which experimental results can differ, and we prime them to understand that they are likely to get a result that fails to line up with something that's expected at some point in their careers. In fact, our data would suggest that that's almost guaranteed. And so if we can change the set of expectations so that we are priming students to expect that they will encounter irreproducibility, the effect on them emotionally might be less, and they might be more willing to consider other types of explanations, like that the original result might be wrong.
Another question here from Samin Vizier, who writes that, oh, excuse me, it strikes me that conflicts with advisors over the failures to replicate is one case of a broader problem, that advisors have almost total control over their trainees' career prospects. And so there are very few avenues for trainees to report problematic supervisors. What do you think universities, departments, professional societies can do to help trainees get out of these labs and into labs with better supervisors, or to even help the students be able to report problematic supervisors so that they don't keep doing harm to future generations of students?
Thanks, Samin. That's a hard one. Yeah, so it's absolutely the case that one of the big problems with the scientific system is that when students become specialized in a particular set of techniques that are so unique to their labs, moving to a different lab when they have a problematic relationship with their supervisor means starting over. It means the loss of several years of research because they need to realign their research skills and interests with that of their new supervisor. I think it's unlikely to assume that we could have clusters of people with expertises so interchangeable that it would make it quite easy for students to move lab to lab. But some programs do have systems in place, for example, of rotations through labs early in one's career so that you can try on both different techniques and different supervisors to get a sense of whether or not it's going to be a good fit and a good relationship. Another thing that I should note about the particular population that we interviewed is we interviewed people who are enrolled in US training grant programs. And this is a program from the National Institutes of Health in the United States that guarantees supported funding for 5 years for the students who are accepted. And when they're in the program, they have a cohort of students in their training grant program, in addition to the cohort of students in their department. And they have a training grant supervisor, in addition to their supervisor and his or her head of department. So the training grant program actually provides a bit of a sort of alternative space for students to go to potentially seek recourse if they have a problematic supervisory relationship, because they can go to the head of the training grant program, not just the head of the department where their PI works. This might incidentally be one of the reasons why when we ask students in the survey about their present state of mental health, using a couple regularly validated tools, we actually found that the mental health of the students that we were interviewing was quite a bit better than other studies have reported about graduate student mental health. And my guess is that one of the reasons why the students we interviewed seemed to have less depression, less anxiety than other studies have shown is because they were attached to training grant programs where the PIs of those programs not only readily volunteered to let their students participate in our study, but in some cases supported the research by doing things like sending out an email to the cohort to ask them to participate. So I suspect here that we've selected actually a group of students who are particularly well supported in a number of ways. And that should actually make us rather concerned in the sense that even though we have interviewed here what I think are probably a cohort of exceptionally well-supported students, they have guaranteed money, they have an alternative route to go, they have an advisor who wanted to participate in a study about reproducibility and mental health. What we still see is that a lot of these students were suffering quite deeply from their early experiences of failure to replicate. So that tells me that students in different circumstances might be impacted even more strongly, if my hypothesis is right, when they encountered a failure to replicate and have a less supportive ecosystem.
Thanks. Now, this is an interesting question. It refers to when we're teaching undergraduates, so very early undergraduates, when we're teaching them sort of fundamental skills and lab techniques. And as part of that training, we often ask students to replicate a technique until they get the expected result as part of skill acquisition. And we only pass the student if they can replicate that lab result, which is typically a very strong effect. So this sets up an early experience of experimenters seeing a failure to replicate as being their fault in these very fundamental tests. What kind of role do you think that's playing here, if any?
Yep, you're 100% right. And that's a very perceptive observation, because if you think back to your own kind of like basic biology or lab training, probably what you did is something that's kind of a toy experiment where you're not actually really investigating a question that people believe we don't know something about. You're taking a finding where the instructor expects that you will get an established result, and then in your lab report expects you to explain why you might have deviated from the result that's expected. So that sets you up to be looking for imperfections in your technique, the expectation that one finding will be sort of stable for all times and all places. And it sort of cements that vision of the world as this algorithmic place where if you do things correctly, the world will respond in one way. Now, there's been some literature from ethnographic studies of the scientific knowledge process that shows that when students get to graduate school, this actually creates something of a kind of crisis moment for them, because all the science that they've been taught in their undergraduate programs is typically based on the sort of most stable, most widely accepted results, things like trying to replicate gravitational constants, using your little, inclined plane or something like that. But when they get to the laboratory, what they encounter is this sort of messy, warm, unsettled frontier of science where far less is known, the results are much more complicated, and where they are expected to make judgment calls about whether or not the experimental system is working. So I think that one thing that we could think about is undergraduate training programs that try and give students real research questions, in that they are expected to find something truly surprising. And there are some types of programs that do this at my own university. And one thing that I would really like to pursue in the future is looking at a comparison between students who are enrolled in the traditional curriculum and this alternative curriculum that exists at my university, to see if they come out with different expectations about how variable the natural world is, how stable the literature is, and whether or not they should anticipate failures to replicate as part of the normal life course of science.
Great answer. The next question is from Harry Collins. There is a paper in the literature that refers to second order measures of skill. So there's a paper in the literature that refers to second order measures of skill that suggests that the science literature should include some indication of how many times the original researcher had to repeat the experiment before they got the result. So how reproducible is it now that they know how to do it, or how long it took them to get there, and so on? Do you think that this would be useful?
Okay, well, Hi, Herrick. Thank you for attending my talk. I'm not familiar with the paper that you're referring to, but I have some thoughts anyway that I think are probably related to what's going on. I think the problem that you're pointing to here is that sort of looping effect that you identified, where researchers judge the skill of a new researcher by their ability to produce the expected result, as in the classic case of the TEA laser. There are other ways, though, that we could try and think about judging experimental skill, or trying to think about which tests are genuinely good tests of experimental skill. So the measure of how many times it took a researcher to get this result might suggest, for example, not only that this is a hard technique, but maybe also that it's a variable technique in some particular way that would mean that we should not use it necessarily as a test of skill. So I'm not entirely sure what an alternative sort of skill testing system might look like, but I think that this is what you're hinting to, is that if we could find some sort of way of assessing skill that wasn't just picking any experimental result and asking the student to replicate it, that we might be able to get a better differentiation between things that are lack of acquired skill and true failures to replicate. Does that answer where you were going?
Another question from Eden Smith. Did the interviewees in your study ever discuss whether they were given opportunities to work as part of teams attempting to replicate prior experiments rather than trying to do it all on their own individually?
Yeah, there was quite a bit of variation in our study in the way that laboratories were set up. So some research groups operated in a very kind of siloed way, where the student was expected to complete their project kind of from the very beginning to the very end without any sort of intervention, as a way of demonstrating their kind of independence as a young researcher. So for the PIs in these labs, what they wanted to see was someone who needed no help from anyone else. That created all kinds of problems, I think, particularly because it was also difficult for those students to then adopt practices that would enhance reproducibility. Think, for example, about blinding or masking. If you want to mask your experimental results, you need somebody else around you who's willing to participate in some way in that labor in order to be able to mask you so that you can do your experiments, or for them to do them for you. And so in a system where students were quite siloed, they had fewer opportunities to sort of see what experiments look like in someone else's hands to get a sense of variation. And they also had fewer opportunities to adopt some kinds of reproducible practices. Now, in other labs, it was quite opposite. One lab that I researched, or that a student mentioned, had a system where they had at least 2 students on any given set of experiments, so that they were always doing internal replications against each other. Now, this sounds like a great system, but it was also one that was not without sort of sociological problems, because what happened in some cases is that one student would fail to replicate the other, find the student's error, and create interpersonal conflict in the lab, where the students were competing with each other to be the better of the pair of experimenters in a way that created a lot of friction. And these implicit hierarchies that emerged within the social structure of the lab through the system of replicating each other then also had negative impacts on scientists developing self-identity. One student told me something along the lines of, "I've never believed that I could be a scientist because of an experience that she had at the very beginning of her career, where her replication partner consistently managed to get more trustworthy results." Now, she had 5 more years of experience under her belt, but that first year experience really stayed with her. So even though having internal replications is great for science, we still need to think about its impact on the scientists, and whether or not we can develop sociological systems that allow people to do reproducible research without compromising the sort of social structure of the laboratory and the researcher's mental health.
I think I might just ask one final question, and it's from me. I'm going to ask you to speculate about interdisciplinary comparisons, and I guess I'm wondering if there's any hope that we can be optimistic that what you've uncovered in this study, in this discipline, is the worst of what we're going to see? Is there any chance that it's better elsewhere?
Well, I think that one thing that is probably quite variable between disciplines is the extent to which researchers believe that a particular technique is dependent on individual skill. So for a lot of the bench scientists that were involved in this study, they very much believe that techniques might be sensitive enough that the way you transferred cancer cells underneath the skin of a mouse might have an impact on how well they would grow. But I think that there are other areas where there's not such a strong presumption that you need skill to get the techniques to work. Other fields might believe that almost all people can quite quickly be competent experimenters. And if there's a stronger belief that the amount of skill is lower, there might be more of an inclination to then believe the contradictory results when they're produced even by a very junior person. So this is just another, kind of speculative hypothesis of mine coming out of this research, is that if we were to try and look at different fields and their assumptions about how much skill was needed to competently perform a technique, you might also see differences in terms of how quickly students trust themselves or their advisors in turn trust them.
Terrific. Thanks very much, Nicole, and thanks to everybody for attending what is obviously a most interesting talk. What I found particularly engaging is that you dealt not just with epistemological implications of reproduction and failure to reproduce, but brought the human aspect of that, the human implications and the human impact of failure to reproduce results. So I think that's a really terrific contribution. So thanks again, Nicole, and thanks to everybody for attending. Those who wish to join in this thanks can do so. Can't do it live, but can use the Q&A and can use the chat to thank our speaker. And we'll finish there. Thank you, everybody.
Thank you.